I'm pleased to introduce one of my colleagues, Dave Williams, who is on the modeling team here at the Allen Institute. And he's going to talk to us about modeling at the Allen Institute for Cell Science. I want to see a bit more of that.
Hello. So the little subtitle is because I think one of the main things that we're trying to do in modeling here is get more information out of the information we've collected. You can see a lot. You can't actually see this much. This is a fake image. But you can see a lot. But there's limitations to how much information we can collect about an individual cell or an individual preparation. One of the obvious limitations that we have is a bandwidth limitation. You only have so much light bandwidth in to excite your fluorophore, and only so much light bandwidth out to observe that excitation. Of course, you can try and put more fluorophores in and just turn up the amount of light you're pumping through the system. But then you get cell death because of phototoxicity. So what are ways that we can see more structures at the same time? What are ways we can get around these limitations? Right now, today, I kind of want to split this into two different approaches. One of which is to try and detect more things in a single measurement, in a single field of view. And the other is to combine multiple measurements of individual cells into one image. So let's talk about that one first. So information can exist in schmootz. I think of schmootz as like the stuff on my glasses, but for the purposes of this talk, let's say it's stuff in images that you don't really know what it means. There's times when there's a lot of structure to an image, but I can't necessarily interpret what's going on in there until I come up with the appropriate transform function. And then all of a sudden, that information is in the right context for me to actually make sense of it. So what's the transform function that we should be applying to our images of cells? Well, there's classic ones. There's ones that are actually physical transform functions. You can introduce dye into the system and see where the DNA is as a result of that. That comes with the limitations of phototoxicity and bandwidth that I talked about earlier. But that structure that the dye is highlighting is actually present in that image, because I can sit down at a laptop, spend some time tracing out where I think nuclei are by hand, and it kind of works. It matches the dye image pretty well. It just takes a lot of time. So is there a way that we can encode the intuition that I have about where nuclei are into a model that can do it faster, cheaper, better? Well, yeah. We can train a model to transform schmootz into information. We can start with the images that we want to transform, sampled and cut down into small segments, and then a ground truth, which is the dye images that we're trying to recreate, and send them through a U-netish neural model. There's more information about this model structure at the website and at the paper describing this work. But briefly, it's just a way of encoding a lot of information that we think is present in an image. And that'll output some kind of image that starts out to be not very good. But because it's a model, you can give it an error function, and it can compare the dye images to the model output images and slowly get better over time. How much better does it get? Well, it works pretty well for nuclear predictions. If this is the bright field image, and this is the dye image, this is the predicted model output image. And it does a good job. It actually denoises this a little bit, because this noise is an artifact of this imaging process rather than something that's present necessarily in the bright field image. This works really well for structures that are dense and show up well in bright field images. So for nucleolus, works great. Again, real dye image, predicted model image. For diffuse structures, or structures that are filamentous, it doesn't work quite as well. So this is myosin 2B, and as you can see, it's a lot more cloudy and doesn't really capture exactly what we want to see. But if we use this system to try and model several structures that we think all show up pretty well in those bright field images, we can combine them and actually see things that we couldn't before. So this is a time-lapse image, and something that wouldn't work very well with a dye-based methodology or a tag-based methodology. And we can see many, many structures in that time-lapse image that we wouldn't necessarily be able to separate or segment before. So that's one approach. Let's talk about combining multiple images of single cells and information we can get from those. So particularly, this is good for cases where that information is not necessarily present in the bright field images. So really what I'm asking here is, can I get a model that's going to tell me the things that I have for some cells, given what I have for every cell? So for all of the cells, I have the cell membrane dye and the DNA dye. For some cells, I have, for every cell, I have one structure labeled, but not every structure for every cell. So really, this is, if I put this in a formalism, it's just saying, what's the probability that I have a given structure image, given an input cell and nuclear image? And again, we can train a model to understand this, with the input being our, at first, just trying to build a model of cell and nuclear shape. So the input is just our cell membrane and nuclear images. And we encode those, we compress them into a small dimensional space, a reduced representation of those structures, and then decode it. This is the same thing as compressing and uncompressing an image. And that gives us our first error function that we use to make the model better. What's the input image minus the output image? But we also want this reduced dimensional space to be kind of smooth because we want to be able to explore it and extract the most likely locations within it. So we take a Gaussian and we say, OK, well, how close is this reduced dimensional space to a Gaussian? And that's our normality error. That's our second error function that we use to train our model. Now, the third thing, and the thing that really gooses this, is you train an adversary that gets to see both the input and the output images and tries to distinguish between the two. If it can, then there's an obvious difference between your images, and you penalize the model. If it can't, then maybe they're pretty close to each other, and that's a good thing. So I'm going to take this part of the model and say, OK, this is our model of cell and nuclear shape. We've got a way to encode them and then decode them. Let's use that and put it into a model of cell, nuclear shape, and structure. That gives us a combined model of cell, nuclear shape, and structure. Same training procedure as before. Now, the clever part is that I can say, OK, well, this is the formalism I have. But I can say, what's the expected structure? What's the most likely structure that I'm going to have given a known cell and nuclear shape? And that can give me a predicted structure for an image where I didn't have that structure before, where I only had cell and nuclear shape. And I can do that for one structure, and another structure, and another structure, and another structure, and then again combine all of those into a single prediction. So let's look at the output of this model. Here's the observed ground truth. These are real cell and nuclear dyes and real tagged structures. These are our inputs to the model, just cell and nuclear shapes. And these are the model output, the cell and nuclear shape combined with the model's prediction of where that structure should be. And it's hard to tell the model predicted ones apart from the real ones. And that is a relatively reasonable measure of how good a model is. So these are two ways that we can combine information and get more out of the images that we already have. I want to thank the modeling team and the rest of the team at the Allen Institute for Cell Science, and of course, the founders. Thanks very much.